So good morning to all of you and welcome back again in our online lectures of Bhalerao Science College, Sauner. So welcome back again all of you dear students of semester 4. So we are discussing here the part of BSc semester 4, paper 2, having the name Genetics and Molecular Biology. In the previous lecture, we have started this paper 2, Genetics and Molecular Biology. As per the protocol and as per the syllabus provided by the RTM Nagpur University, paper 2 consists of total 4 units. Out of these 4 units, unit 1 will be allotted to me. And this unit 1, it consists of the part of Mendelism and gene interaction and the linkage. In the previous lecture, we have started the Mendelism that Gregor John Mendel was one of the famous personality in the field of genetics. That Mendel introduced one experiment, hybridization experiment. With the help of this hybridization experiment, he gives information that how the characteristics, basically the genetic characteristics, are transferred from one generation to another generation. The transmission of these characteristics, which is a part of inheritance, and inheritance can be done with the help of specific factor or the specific material. This was provided by the Gregor John Mendel. Gregor John Mendel was a methodological person. He performed experiment with the help of specific methods and stepwise procedure. With the help of stepwise procedure, he performed the experiment, hybridization experiment on the garden pea plant. In the previous lecture, we have discussed some of the introductory topic related to this Mendelism. That is a part of introduction or the biography of Mendel. Mendel was coming from the very poor farmer family, and he completed his graduation from the University of Vienna and started to do the job as a priest and as well as a teacher. And then he take education also from the University of Austria, and we find out some important techniques related to this hybridization experiment at which University of Vienna and University of Austria. He performed experiment on the garden pea plant having the scientific name Pisum sativum, and also we have discussed that why he choose only the garden pea plant. That garden pea plant, it is herbaceous plant. It is an annual plant. It can complete its life cycle within a short period of time. It consists of very much different contrasting characters. इसमें बहुत सारे contrasting characters थे. Then it is a self-pollinating plant. That's why he chose the garden pea plant for his experiment. From this garden pea plant, he selected the 7 different contrasting characters to perform the experiment. For example, length of the stem height, and then seed shape, seed color, pod shape, pod color, flower position. These are the 7 characteristics selected by the Mendel for his experiment. Then Mendel's experiment, it having some important steps, or we can call it as a procedure of Mendel's experiment. The procedure of Mendel's experiment, it consists of mainly some of the important steps. For example, the selection of parents and obtaining their pure lines. Then cross between artificial cross, the hybridization experiment along with the selected parents. Selected parents के बीच में क्रॉसिंग करना, और क्रॉसिंग करने के बाद हमें वहां पे मिलने वाली थी F1 generation. Then after that he go for the F2 generation. So the procedure of Mendel's experiment, it consists of the selection of parents. Firstly parents select करते थे. उसके बाद में प्योर लाइंस सिलेक्ट की जाती थी. उसमें प्योर लाइन फॉर एग्जांपल प्योर टॉल, प्योर ड्वार्फ, प्योर येलो राउंड, प्योर ग्रीन विंकल. Then they go for the crossing, artificial cross or the hybridization, which produce the F1 generation, and selfing of this F1 generation, which produce the F2 generation. So finally, by performing the basic steps of this Mendel's experiment, at least he find out that something is transferred from one generation to another generation. So up to this, we have discussed in the previous lecture, which is a part of heredity and variations. Means William Bateson, an Englishman, gave the name this developing science as a heredity and variation as a genetics in 1905. He coined the term genetics from the Greek word meaning to generate. So William Bateson just introduced the term that is genetics. And basically genetics, it is the scientific study of heredity and variation. If you go through the scientific study of heredity and variation, it is called as a genetics. This genetics term was introduced by the scientist William Bateson, but the scientific explanation related to this genetics was given by the Gregor John Mendel. That's why the Gregor John Mendel was popularly known as a father of genetics. Now let's just talk about this today's topic, that genetic terminologies or the symbols which are utilized in the Mendel's experiment. So Mendel was very methodological person. He performed the experiment with the stepwise procedure, and each and every condition or each and every steps performed very carefully by that Mendel. So before we going to again discuss in deep the Mendel's experiment, we should know some important terms or the terminologies which are utilized or which can be utilized in the Mendel's experiment. While understanding the and expressing the basics of genetics, it is at most essential to understand the basic terminology commonly used. Some of these are as follows. मीन्स कुछ जो इम्पोर्टेंट टर्म्स है वो हमें समझना बहुत ही जरूरी है टू परफॉर्म द मेंडल्स एक्सपेरिमेंट. ठीक है, मेंडल का एक्सपेरिमेंट परफॉर्म करने के लिए या फिर मेंडल के एक्सपेरिमेंट में हमें डीप स्टडी करने के लिए ये सभी बेसिक टर्मिनोलॉजीज हमें पता होनी चाहिए. ठीक है. These basic terminologies may ask in your examination, theory examination, especially for the question number 5, last question के लिए answer in one sentence. आप लोगों को एक ही सेंटेंस में उस टर्म को एक्सप्लेन करना होता है या फिर आंसर करना होता है. It is for 1 mark. So very short answer question. These terminologies may ask in that question number 5. So let's just take a look regarding this terminologies which are utilized in the Mendel's experiment. While understanding and expressing the basics of genetics, it is at most essential to understand the basic terminology commonly used. Some of these are as follows. The first terminology, that is alleles. A gene controlling any character has a 2 or more than 2 alternative forms. However, in normal case, each gene has a 2 alternative dominant and recessive forms, develops contrasting character known as alleles. So finally, we can say that the 2 or more than 2 alternative forms. Alternative forms means it may be a dominant or it may be a recessive form which is present on a gene. The 2 or more than 2 alternative forms which are present on a gene, it may be a dominant or recessive. It is called as alleles. उसे हम लोग alleles कहेंगे. Let's just talk about the next terminology: dominant allele and a recessive allele. What is this dominant allele? An allele which expresses itself by producing its character controlled by it in the heterozygous or homozygous dominant state. Such an allele is known as a dominant allele. So the allele which express themselves in the next generation, जो allele खुद को express कर रहा है next generation में, उस allele को हम लोग dominant allele कहेंगे. Recessive allele, the allele in the presence of dominant allele not express itself in the heterozygous state or expressed in the homozygous recessive state. Such allele is known as a recessive allele. So the allele which does not express themselves even in the presence of that dominant allele or the dominant character, it is a part of recessive allele. Now let's just talk about this gene. It is a very easy definition of gene. As per the classical genetics, the genetic factor which control the specific character in an individual. मतलब क्लासिकल जेनेटिक्स के हिसाब से अगर देखा जाए तो जीन का मतलब होता है, ये एक फैक्टर होता है, जेनेटिक फैक्टर होता है कि जो पर्टिकुलर कैरेक्टर को कंट्रोल करने के लिए यूज किया जाता है. सो जीन बेसिकली इट इज अ जेनेटिक फैक्टर व्हिच कंट्रोल द स्पेसिफिक कैरेक्टर इन एन इंडिविजुअल. किसी भी इंडिविजुअल में कैरेक्टर कंट्रोल करने का काम ये जीन करता है. एज पर द मॉडर्न जेनेटिक्स के हिसाब से अगर हम देखते हैं, जीन इज नथिंग बट द सीक्वेंस ऑफ डीएनए which codes for a single polypeptide or RNA molecule, for example rRNA and a tRNA. Basically, the gene, it is a sequence of DNA. We can also call it as it is a segment of DNA. Sequence मतलब जो न्यूक्लियोटाइड sequences present रहेंगे DNA पे, adenine, guanine, thymine, cytosine in the form of this sequence. तो ये sequences से बना हुआ एक structure अगर हम लेते हैं DNA का, that sequence of that particular DNA, it can be considered as a gene. Now, homozygote and heterozygote. Now, what is this homozygote? An individual may have two copies of the same alleles. Such an organism is called as homozygote, and such a condition is known as a homozygous condition. For example, capital A, or small a, small a. So that individual, it consists of the two copies of the same alleles. Means the similar alleles. If the similar alleles are present in case of the particular individual or organism, किसी ऑर्गेनिज्म के केस में या फिर इंडिविजुअल के केस में अगर सिमिलर अलील्स प्रेजेंट होते हैं, then this condition is called as a homozygote or the homozygous condition. ठीक है, उसे हम लोग homozygote कहेंगे या फिर homozygous condition कहेंगे. So this homozygote or homozygous condition, it is a part of similar alleles. Now let's just talk about this heterozygote. Individual may have two different alleles of the same gene, then it is referred as to heterozygote, and that condition is shown by the individual is termed as a heterozygous individual. For example, capital A, small a. So if we go through the comparative manner, homozygote condition, it is a capital A, and small a, small a. And heterozygote condition, it shows the different alleles of the same gene. For example, capital A and small a. It is a heterozygote or the different alleles of the same gene. Now let's just talk about this phenotype and genotype. The appearance of an individual with respect to the specific character or all of its character includes the morphology, biomolecules, etc. is referred as a phenotype. So this morphology word is very important. Morphological characters means we can say that the external characteristics of that particular organism. So on the basis of external characteristic, we can consider the appearance of that particular organism. Means on the basis of external characteristic, for example, that particular person if having the larger height, we can call it as a tall. So on the basis of just external features के basis पे हम लोगों ने वहाँ पे character designate किया या फिर identify किया, and this condition is called as a phenotype. Phenotype basically it is related to the morphological characteristic of that particular organism. Now genotype, the genetic makeup of the cell or organism. In other words, gene or the kinds of alleles of specific gene constitute the genotype of an organism. So on the basis of genetic characteristics, we can identify the different characteristics of that particular organism. So it is called it as a genotype. If we compare this genotype with the phenotype, genotype, it is basically the internal characteristics, and a phenotype, that is the external characteristics or the morphological characteristics. Test cross and back cross. The cross between F1 hybrid and the parent with the recessive alleles for that character in a question is known as a test cross. Test cross means if you go through the crossing between the two pure parents, then we will get the F1 generation. अगर हम लोग दो प्योर पेरेंट्स के बीच में क्रॉसिंग करते हैं तो हमें वहां पे एक नेक्स्ट जनरेशन मिलती है. दैट नेक्स्ट जनरेशन इज कॉल्ड एज द F1 generation. So you can see here the smooth variety and the wrinkle variety. If we go for the crossing between these two parents, capital S and small s, we will get the next generation capital S, small s. This capital S, small s, it is called as the F1 hybrid or the F1 generation. Now what is this test cross? The cross between F1 hybrid and a recessive parent, right? Recessive means smallest, capital S, smallest, cross with smallest. So this is the basic condition behind the test cross. The test cross, it is the cross between the F1 hybrid and with the recessive alleles for that character is in question is known as a test cross. उसे हम लोग test cross कहेंगे. What about this back cross? Back cross के बारे में पढ़ेंगे. The cross between F1 hybrid and any one of its parental individual referred as a back cross. So back cross, it is a condition in which the F1 hybrid is crossed with any one of the parent. It may be a dominant parent or it may be a recessive parent, and that type of cross is called as a back cross. Now, what is this progeny? Members of the same biological family with the same mother and father, the offspring from a given mating. जो ऑफस्प्रिंग हमें मिलने वाला है नेक्स्ट जनरेशन में, जो ऑफस्प्रिंग मिलने वाला है, ठीक है, या फिर जो मेंबर मिलने वाला है बाय द क्रॉसिंग बिटवीन द मदर एंड फादर और क्रॉसिंग बिटवीन द टू प्योर पेरेंट्स, then that next individual or that next offspring is considered as a progeny. Now, hybrid, the progeny produced by the crossing of organism that exhibit different inherited traits are called as a hybrid. हाइब्रिड सब लोगों को पता है, दो प्योर पेरेंट्स के बीच में क्रॉसिंग करने के बाद डिफरेंट कैरेक्टरिस्टिक के साथ, मतलब वो ना मदर की तरह रहेगा ना फादर की तरह रहेगा, ठीक है, वो डिफरेंट ही वैरायटी वहां पे प्रोड्यूस हो जाएंगी. दैट इज द केस ऑफ हाइब्रिड. नाउ व्हाट इज दिस मोनोहाइब्रिड क्रॉस एंड डायहाइब्रिड क्रॉस? Mono means single and di means two. Means you can consider here mono means at a time they take only single characteristics. And what about this di means two characteristics at a time. So the cross between the pure parents differing in only one trait, or the cross in which only one trait is being considered, and it will be responsible for the production of new generation or the F1 generation, is called as a monohybrid cross. Means if we cross between the two parents, दो parents के बीच में crossing करते हैं, which are responsible for only one trait, ठीक है, single character के लिए. For example, one parent is tall and another parent is dwarf. So this is a single character, that is a height, right? Height, it is a single character, but it is contrasting. So by considering the single characteristic, if we go for the crossing, then we will get the next generation, that is F1 hybrid. It is called as a monohybrid cross. So हम लोग monohybrid cross. So the cross between the two parents differing in their single characteristics or single pair of contrasting character to produce the F1 generation, it is the case of monohybrid cross. Dihybrid cross, the cross between the parent differing in the two traits. यहाँ पे single trait था, only one trait, और यहाँ पे दो traits है. The cross between the parent differing in the two traits, or the cross in which the two traits are considered for studies, it is considered as a dihybrid cross. Means here we can take the two characteristics at a time. यहाँ पे हम लोग दो characteristic at a time लेने वाले हैं dihybrid cross के केस में. Now the dihybrid in an individual genotype characterized by the heterozygosity at the two loci. In other words, the progeny form from a cross between the two homozygous parents differing for the two genes or the characteristics. So if you go through this point of the dihybrid, in individual genotype characterized by the heterozygosity, मतलब अगर सिंगल इंडिविजुअल को हम कंसिडर करते हैं तो हेटेरोजाइगोसिटी शो करता है at the two loci. Loci means it is coming from the word locus. Locus means position. The position on the gene. Gene पे उन alleles की क्या position है? Two different alleles बोला, two different position बोला है उन लोगों ने. In individual genotype characterized by the heterozygosity at the two loci. In other words, the progeny from a cross between the two homozygous parents differing for the two genes or the characters. मतलब दो पेरेंट्स रहेंगे यहाँ पे, two parents will be there. Two parents will be there, but they show the different characteristics. For example, we can take the two characteristics at a time. For example, yellow color and round shape, yellow round, and a green wrinkle. So these are the two characteristics belonging to the same gene. So it is a case of dihybrid condition. Now monohybrid condition. An individual that is heterozygous for a pair of alleles at a locus under study. Locus means position. मैंने आप लोगों को बताया एक ही जगह पे वहाँ पे सिर्फ एक ही कैरेक्टर रहेगा वहाँ पे. So it is a case of monohybrid condition. Now the first filial generation and the second filial generation. It is a case of F1 generation and a F2 generation. So let's just talk about this first filial generation. Filial stands for, it is a Greek word, filial stands for son or the offspring. First filial generation, F1 generation. The offspring resulting from the first experimental crossing of the plants or the animals and denoted by the F1. जो ऑफस्प्रिंग हमें क्रॉसिंग के बाद मिलता है, ठीक है, फर्स्ट क्रॉसिंग के बाद, फर्स्ट क्रॉसिंग करने के बाद हमें जो ऑफस्प्रिंग मिलता है नेक्स्ट जनरेशन में, दैट ऑफस्प्रिंग इज कॉल्ड एज द F1 generation or the F1 offspring. It is also called as a first filial generation. Filial, it is a Greek word coming from, it is meaning that the son or offspring. Similarly, the second filial generation, the progeny which is produced by intercrossing or the self-fertilization of F1 individual and denoted by F2. जो प्रोजेनी हमें F1 जनरेशन की सेल्फ फर्टिलाइजेशन करने के बाद मिलते हैं, self fertilization we can call it simply as a selfing. Selfing means the cross between F1 generation into F1 generation. Means after getting the F1 generation, we go for the F2 generation, and this F2 generation can be obtained by the crossing with F1 generation with F1 generation. Now, Hemi Zygote, a diploid organism having one copy of a gene in place of normal two. Hemi zygote के केस में, a diploid organism. Diploid means the two sets of chromosome. For example, 2n is equal to 46. A diploid organism having the one copy of gene. Organism diploid रहेगा लेकिन उसके पास में single copy रहेंगे, one copy of gene in place of the normal two. ठीक है, normally two copy की जगह पे वहाँ पे सिर्फ एक ही copy रहेंगे gene की. That is the case of hemizygote. For example, in case of sex linkage and in deletion heterozygous, a gene present in a single dose or the copy. Now the Mendel's laws of inheritance. Let's just talk about this Mendel's laws of inheritance. After performing the Mendel's experiment, Mendel gives the three important laws. Here they explain only the two important laws: law of segregation and law of purity of gametes. And the second one is the law of independent assortment. So after performing the Mendel's experiment, Mendel gives some important laws related to the inheritance in the transmission of character from one generation to another generation. Basically, the three laws, that is a law of dominance, which we have studied in the previous lecture. That is a part of law of dominance. After performing this monohybrid cross, Mendel's experiment. In this Mendel's experiment, after performing the monohybrid cross, we can find out the law of dominance. Means according to the law of dominance, whenever we go for the crossing, if we go for the crossing of the two pure parents, then we get the F1 generation. In this F1 generation, the phenomena by which one trait appeared is called as a dominance, and which other is which does not appear in the F1 generation is referred as a recessive. So this is the case of law of dominance. Means after performing the monohybrid cross, we can consider that one of the character is dominant character in the F1 generation, and another character is a recessive character. So it is a case of first law of Mendel's experiment, that is the law of dominance. Here they explain only the two laws, that is the law of segregation, or it is also called a law of purity of gametes. And second one is the law of independent assortment. Let's just talk about this Mendel's laws of inheritance. On the basis of brilliant experiment performed with the Pisum sativum, that is a garden pea plant, and analysis of F1 and F2 data for the different traits or the characters using the statistical methods, Mendel formulated the fundamental laws of heredity. So according to the Mendel's experiment, right? That is, Mendel was very methodological and it's statistical this procedure following person. So that his experiment was considered as a brilliant experiment. This brilliant experiment performed on the Pisum sativum. We know that all that Pisum sativum having the scientific name of the garden pea plant, and analysis of F1 and F2 data of the different traits or the characteristics using the statistical methods. Means in this Mendel's experiment, Mendel used statistical methods to calculate that data and analyze that data and find out the result. So to find out the result and perform the experiment, Mendel performed these statistical methods, and this condition was not supported by that previous or the famous scientist at that time. That's why the Mendel's experiment was rejected. But later on, the three scientists Hugo de Rice, Correns, and Schermack, they rediscover the Mendel's law and they know the greatness of Mendel. So by using the statistical methods, Mendel formulated the fundamental laws of heredity. मेंडल ने जैसे ही वहां पे डेटा कलेक्ट किया, then he go for the statistical methods and he find out the two important laws. Law of segregation, basically it is the second law of Mendel's experiment, which is also called as law of purity of gametes. And next one, that is law of independent assortment. First law, let's just talk about this law of segregation or law of purity of gametes. Mendel proposed the first fundamental law as the law of segregation, which can be explained more clearly by considering the following assumptions. मेंडल ने जो लॉ दिया था, फंडामेंटल लॉ जो दिया था, लॉ ऑफ सेग्रीगेशन, इसको एक्सप्लेन किया था उन्होंने कुछ इंपॉर्टेंट assumptions के बेसिस पे, कुछ इंपॉर्टेंट पॉइंट्स के बेसिस पे. The first assumption related to this law of segregation or law of purity of gamete is each character is produced by the specific gene. हर एक कैरेक्टर जो होता है वो एक स्पेसिफिक जीन से प्रोड्यूस किया जाता है. Dominant alleles and recessive alleles are the alternative form of a gene. मतलब इन्होंने सबसे पहले बताया कि हर एक कैरेक्टर जो होता है वो स्पेसिफिक जीन प्रोड्यूस करता है. मतलब ऐसा नहीं होता है वहां पे कि दोनों पेरेंट्स के कैरेक्टरिस्टिक जो होते हैं कंबाइन हो जाएंगे उसके बाद में एक्सप्रेस होंगे, ऐसा नहीं होगा. हर एक जीन उसका कैरेक्टर सेपरेटली एक्सप्रेस करेगा. वहां पे डोमिनेंट अलील्स और रेसेसिव अलील जो रहेंगे उनके पास में अल्टरनेटिव फॉर्म्स रहेंगे उनके. Development of contrasting character is governed by the two alleles, dominant and recessive of the gene. मतलब जो कॉन्ट्रास्टिंग कैरेक्टर्स आप प्रोड्यूस होंगे, टॉल वर्सेस ड्वार्फ, येलो वर्सेस ग्रीन, राउंड वर्सेस रिंकल, these contrasting characteristics will be governed by the two alleles. Means the two different alleles will be there, separate alleles basically they are there to express the particular characteristics. Means there will be a no mixing of characteristics. An individual's somatic cells having the two copies of each gene, one from the parental, that is a male, and other from the paternal and maternal that is from the female. So finally they say that the characteristics does not mix here, they just combine and they express independently here in the Mendel's experiment of the law of segregation.